This is Group 8 and I'm here to present you with chloramphenicol.
Yeah, tell me, how does chloramphenicol look like?
This is the structure of chloramphenicol. As you can see, it has two chiral centres, located at the C1 and C2 alcohols, both of which have R conformations. This conformation is important for activity, as is the aryl structure. The para-nitro group, however, can be replaced with other functional groups. The molecule will still have activity, just not as high as the activity of chloramphenicol. As you can tell by these properties, chloramphenicol fits Lipinski's rule of 5. It means that it's perfectly fine to be administered orally. However, it can't be infused because it has a pKa of around 7.49. By converting the alcohol group closest to the amide into an ester with a carboxylic acid, chloramphenicol succinate is made. The pKa becomes 3.68, so the solubility is greatly increased. Chloramphenicol is the first-line treatment for superficial eye infections, and is the second-line treatment for bacterial infections in the external ear canal, typhoid fever, and life-threatening infections caused by Haemophilus influenzae. Eardrops, eye ointments, and eye drops are used to treat ear and eye infections. Solutions for injections or infusions are only used in life-threatening situations.
I see. So can you tell me more about the side effects of chloramphenicol?
Several side effects of chloramphenicol come from systemic administration. One of them is bone marrow suppression. This can be reversible or irreversible. In irreversible bone marrow suppression, chloramphenicol can cause aplastic anemia, fatal pancytopenia, leukopenia, and thrombocytopenia. The exact mechanism of this is uncertain at the moment. It is suspected that chloramphenicol-induced aplastic anemia is related to DNA damage caused by metabolites in bone marrow. Reversible bone marrow suppression is dose-related and predictable. Red blood cells are suppressed inside the bone marrow. It's probably due to the inhibitory action of chloramphenicol on mitochondrial protein synthesis. This occurs when chloramphenicol plasma concentrations are 25 micrograms per milliliter or higher. This is often observed in patients who are undergoing large doses of chloramphenicol or prolonged treatments, and in some cases, both. Another severe side effect is grey baby syndrome. The manifestations in the first 24 hours are vomiting, refusal to suck, irregular and rapid respiration, abdominal distension, periods of cyanosis, and passage of loose green stools. Soon the babies become flaccid and hypothermic. At last, the most characteristic symptom of the syndrome is that the babies develop a grey or blue skin colour due to poor blood circulation. This syndrome is caused by excessive exposure or prolonged doses of chloramphenicol in newborn babies, especially if they're premature. The underlying reason why chloramphenicol can cause this is that the chloramphenicol is metabolized through phase 2 metabolism by undergoing a conjugation reaction in adults. This reaction is aided by UDFP glucuronate, an enzyme which aids in the addition of a glucuronic acid molecule to the indicated hydroxyl group, resulting in chloramphenicol glucuronide. The metabolites can then be excreted in urine or bile. However, this reaction doesn't take place in babies, so that the drug cannot be excreted from the body. This means that it accumulates and eventually reaches toxic levels in the liver.
I see. I feel like I know everything about chloramphenicol already.
Right. Tell me how it's manufactured. There are many methods of manufacturing chloramphenicol, and research continues on its synthesis. One of the methods that is used commercially in Western Europe was developed in the 1960s and 1970s. This method involves condensation of benzaldehyde using 2-nitroethanol in basic conditions to produce nitrodiol. The nitro group in nitrodiol is reduced to 2-amino, 1-phenyl, 1,3-propanediol, which is then acetylated using acetic anhydride, for which pyridine is a catalyst. The acetylation product is nitrated using concentrated nitric and sulfuric acid, and the product of this is then hydrolyzed to remove acetyl groups. Through chiral resolution, the racemic mixture is separated and the stereoisomer is acetylated with dichloroacetyl chloride at the amine to produce a high yield. Thanks to these techniques, we don't need to spend a lot of time culturing bacteria to produce chloramphenicol.
Bacteria culture? Why?
Chloramphenicol was first discovered in 1947, where it was extracted from cultures of Streptomyces venezuela in soil. The samples were collected from a mulch field in Venezuela and from a compost soil sample of a horticultural farm in Illinois. In 1948, chloramphenicol was incorporated into clinical practice under the trade name chloramycetin. Chloramphenicol became the first antibiotic to be manufactured through large-scale chemical synthesis. At this point, the mechanism behind chloramphenical's antibiotic properties weren't clear yet.
So, is it clear now?
Yeah. Studies show that chloramphenicol targets bacterial cells exclusively, particularly the ribosome. The 50S subunit of bacterial ribosomes is reversibly bound to chloramphenicol. This changes the conformation of peptidal transferase in ribosomes, so peptide bond formation is inhibited. The mechanism relies on different functional groups. First, the hydrogen bond donors and acceptors form hydrogen bonds with various nucleotides of the peptidal transferase cavity, such as the ones pointed out here. Secondly, the phenol group form pi-pi stacking interactions with the cytosine ring at the cavity here. In addition to these, putative cations MgC1 and MgC2, a mediator, further stabilize the bonding between chloramphenicol and the peptidal transferase cavity by facilitating the bonding of the 3' hydroxyl group and the para-nitro group to the peptidal transferase cavity. Certain aspects of chloramphenicol are still unclear. This is why research on chloramphenicol has never stopped.
Wow! Can you tell me a bit about the development history of chloramphenicol?
Great. Let's start in 1948. American medical researchers Theodore Woodward and colleagues published the first paper on the use of chloramphenicol to treat typhoid fever. It revolutionized the field, reducing both morbidity and mortality. Unfortunately, later on in the same year, chloramphenicol was linked to rare cases of aplastic anemia and fatal blood dyscrasias. Different derivatives were developed in the hope of offsetting these severe side effects. In 1952, thiamphenicol, a structural analogue of chloramphenicol, was first synthesized. The nitro group was replaced with a methynyl sulfyl group in thiamphenicol. It does not cause fatal aplastic anemia, but causes reversible toxicity in bone marrow and kidneys. Its antibacterial activity is weaker than that of chloramphenicol. This is why thiamphenicol is primarily used in veterinary practice now. By the time in the early 1960s, more than 1,000 cases of severe bone marrow aplasia were attributed to the use of chloramphenicol. As a result, chloramphenicol usage was placed under restrictions. This, however, didn't stop the research because of the rise in antibiotic resistance. Chloramphenicol was discovered in the early 2000s. It's synthesized by replacing the position-free hydroxyl group of chloramphenicol with a fluorine atom. Chloramphenicol was found to be broad-spectrum antibiotic in vitro. The antibacterial activity is as strong as chloramphenicol, but it also combats chloramphenicol-resistant strains and has milder side effects. In vivo, chloramphenicol was reported to penetrate well into the cerebrospinal fluid, showing promising pharmacokinetic properties to treat meningitis. However, chloramphenicol cannot deal with strains bearing resistant genes of drug exporters and mutators. It is expected that chloramphenicol can be extended to clinical trials in the near future. Clear?
Yes. So to conclude, chloramphenicol is a broad-spectrum antibiotic that works by inhibiting peptidal transferase in bacterial ribosomes. It is the first antibiotic to be fully chemically synthesized at an industrial scale. It is the first-line treatment for superficial eye infections and bacterial ear infections. The possible side effects of chloramphenicol include the gray baby syndrome and bone marrow suppression.